Well, good morning then. Hi, I'm Chris Fanta, and it's my pleasure to welcome you to Partners Asthma Center's Asthma Grand Round. And for those of you joining us virtually online, which is everyone now and likely for the rest of the academic year, I want to remind you that you have the opportunity to submit questions to our speaker today by texting me at 617-513-6043. Please write that number down. And then for CME credit, if you wish, just email me your name and degree, and we'll submit your request to Harvard Medical School for CME credit. And I hope you don't have any problems, but if there are technical problems, there's this wonderful email address that you can use, bwhwebcasttroubleshooting@partners.org. So it's my great pleasure to introduce our speaker today, Dr. Nora Barrett, who's trained as an allergist and a pulmonologist. And although we in the Pulmonary and Critical Care Division would have loved to have claimed her for our own, she is in fact a member of the Allergy and Clinical Immunology Division here at the Brigham and Women's Hospital and director of their research training program. And as many of you already know, and the rest will soon discover, any day when you get to learn from Dr. Barrett is going to be a good day. And I'm particularly eager about today's program and eager to hear, and I'll tell you, just let me tell a very brief anecdote about why. It was not that long ago I was in my office, and in walked one of my mentors, brilliant mentors, who said, Chris, so what do you think is the important cell in asthma? The key, if you were to invest your research dollars, what cell would you study? And I'm racking my brain, what's the right answer? Is it the mast cell? Is it the eosinophils? Maybe it's helper lymphocytes? And I didn't know, I'm not sure what I offered, but I asked of my wise mentor, what do you think? And the response was the epithelial cell. And so today we're going to hear from Dr. Barrett about reprogramming the epithelial cell in type 2 inflammation. Dr. Nora Barrett.
Great. Thank you so much for that introduction, Chris. So I'm going to, seeing as we're in an empty room, take my mask off, and hopefully we'll broadcast clearly at home. So this is going to be a little bit of a departure today from sometimes what we see at the Partners Asthma Center Grand Rounds. It's going to be the infusion of basic science. So although it might not always be practical for your clinics right now, I'm hoping at least it will be thought-provoking. So I don't have any disclosures that relate to any information that's going to be talked about here. So we're going to take a brief history of the airway epithelium and kind of what our opinion has been about it in research, and why I'm so passionate and excited about studying it going forward, and think it has so much potential for helping us to understand pathobiology in our patients and direct therapeutic targets. And implicit in this is we're going to refocus, although I am trained in allergy immunology and we've of course spent a long time studying innate immunity in asthma from the perspective of the hematopoietic inflammatory cells. Today we're really going to focus on how some of those cells talk to epithelial cells, reprogram them, and how in kind of a feed-forward loop one might sustain chronic inflammation in a couple of ways. So I'm going to start with what we've known about asthma for 20 plus years, and that is that asthma is, of course, a disease of airway inflammation and airway remodeling. The most clear pathobiologic mechanisms are those of type 2 immunity. And we would say that in the year 2020, type 2 cytokines, which include IL-4, IL-5, IL-9, and IL-13, are made not just by Th2 cells, but also by a host of other tissue-resident cells, including innate lymphoid cells, the type 2 subset, and tissue-resident mast cells. Of course, type 2 cytokines have many different kind of pleomorphic functions, but each of them has functions that are stronger for one particular cell type or another. So for instance, IL-4 is a very, very important cytokine for the generation of IgE and for class switching in B cells to make IgE-producing plasma cells. IL-5 is an incredibly important growth factor for eosinophils and helps to grow them from the bone marrow and get them into the bloodstream. IL-9 that's here is an important mast cell growth factor. And IL-13 has stood out among this group for a long time because of its effect on stromal cells. We know that IL-13 added to cells in a dish or overexpressed in mice can cause airway smooth muscle hypertrophy. It can cause extracellular matrix deposition, and of course can cause goblet cell metaplasia, the kind of hallmark feature of immune imprinting on the epithelium that we're all familiar with. How this plays out when we look at the structure of the lung in severe asthma, kind of some of the most canonical pictures, you can see these large bands of airway smooth muscle that are hypertrophic over here. You can see extracellular matrix deposition, so this thickened basement membrane here. And you can see goblet cell metaplasia, which is stained here with Alsatian blue. So sometime in the last 20 years, we've had a couple hints that the airway epithelium might not just be this passive structure that's imprinted by the immune system, but actually might have pathobiologic molecules that are actually important in eliciting airway inflammation. This has come both from mouse models, which I won't review here today, and also from GWAS studies. Here is just a retrospective review of some of the top hits in GWAS studies in asthma, and what I'd like to highlight here in some of the red are cytokines or other structural molecules that are highly expressed in airway epithelium. Variants at these sites increase the risk of developing asthma. So from both murine models and GWAS studies, we have the idea that the airway epithelium might be a little bit more dynamic and may play a very pathobiologic role in inflammation. And I would say by about 2015, we have this beautiful review from John Fahey in Nature Reviews Immunology, discussing and summarizing our thoughts about that, and that is that airway epithelial cells make many products, including 3 hallmark cytokines, TSLP, IL-25, and IL-33. These come from the structural cells, not likely from goblet cells, and that these have very important afferent immune functions. So on dendritic cells, TSLP can act at the TSLP receptor on DCs and upregulate a very important molecule called the OX40 ligand. When the activated dendritic cell then goes to a lymph node, a cognate interaction here can help stimulate a naive T cell to one of multiple fates, including developing to be a T follicular helper cell that causes antibody secretion, and a Th2 cell. These molecules can also amplify memory Th2 cell cytokine generation, which is shown here, and also can have really important influence on activating innate immune cells. And in these cells, TSLP, IL-25, and IL-33 can drive type 2 cytokine production from most of these cells in the absence of any antigen-specific effect. So fast forward just a few years later, and we have the advent of a technology that has really changed our thinking about the airway, and this is single-cell RNA sequencing. Shown in these plots, which are called Tisney plots, are biopsies. This is of the asthmatic bronchi on the left, and on the right you see on the sinonasal mucosa of patients with chronic rhinosinusitis. Each dot here on these maps of these surgical resections are the transcriptome of a single cell, and the similarly colored clusters are transcriptionally similar cells and define airway epithelial cell populations, at least at the transcriptional level. What you can see, I hope, here on the left is, unlike finding that there are ciliated cells and there are goblet cells, that there's actually an incredible diversity of airway epithelial cells. Included are ciliated and the goblet, but also the club cells, which we were well aware of, an unusual hybrid state, which is called a mucus ciliated cell that has now been reproduced by other labs, submucosal gland cells, and an unusual and newly described cell population called the ionocyte. Over here on the bottom half of this picture shows 4 populations, transcriptionally distinct populations of basal epithelial cells. These cells are airway epithelial progenitors. They're a stem-like cell, and most of the differentiated airway epithelial cells derive from these. Shown here on the right is something similar. These are airway epithelial cells from the biopsies of the patients with chronic rhinosinusitis. And again, I haven't colored them in distinct colors, but you'll see each number here represents a transcriptionally distinct cluster. These biopsies, however, are colored over based on the patient of origin or disease of origin. So what you can see here is that patients with polyp disease have certain transcriptional states in their epithelial cells, and those are quite distinct from those who have CRS without polyposis. So highlighting not only the diversity but the plasticity of airway epithelial cells and how we have to think about them. So I'll just want to introduce briefly now how we think about the airway epithelium, which is a really amazing diversity of cells. We have these basal progenitors, which are shown here in line the basement membrane. They are the primary site of IL-33 and TSLP production in the airway epithelium. We have these individual cells here called ionocytes. We're not going to get to talk too much about them today, but these were recently discovered and made famous because they're the dominant site of CFTR expression. They probably have important roles in regulating air surface liquid and solute concentration, and they also produce some immune molecules, such as the one referred to here, coquelin, which have important antibacterial properties. We have recently discovered there are groups of neuroendocrine epithelial cells, and through their production of certain neuronal peptides like GABA and CGRP, these cells can elicit airway inflammation. And we also have a class that we'll talk a little bit more about today called brush, tough, or solitary chemosensory cells, which make IL-25, another one of the triumvirate of epithelial cell cytokines, in addition to some molecules that are close to my heart, the Cysteinyl leukotrienes, pro-inflammatory lipid mediators that are derived from arachidonic acid. Then over here we have on the right goblet cells, and of course they secrete the mucins, including MUC5AC. So quite a diversity of cells to consider. So I'm going to spend some time focusing on two distinct cell types and review some recent basic science from our group on that, and then we'll bring it back at the end to understand where we think these areas may be going. So the first one is focused on brush cell expansion and function in what I'm calling T2I, which is, of course, type 2 inflammation. So brush cells come by many names. They're called tuft, brush, or solitary chemosensory cell family. They are found in the GI tract, in the airway, in the nasal mucosa, and interestingly in the olfactory mucosa, which is up near the sphenoid sinuses. They're found in other mucosal sites too, the pancreas, urethral glands, whatnot, but in the airway where we see inflammation, and in the GI tract where we see type 2 inflammation, these are the dominant places that have been studied. This is what a tuft cell or brush cell looks like. It has an amphora kind of shape to it, or a Greek urn shape, and it was identified histologically some time ago based on these microtubules that form the apical villi and are displayed over here. You can see that these cells have some unusual features. They have lateral projections that in some tissues extend in towards the nuclear membrane of an adjacent cell with kind of unknown function, but of course is tempting to hypothesize whether there can be transfer of nuclear materials. These cells are characterized by a couple of other very unique things, and one is they express G protein-coupled receptors that are usually found on taste buds. So taste buds are groups of cells that are closely grouped. These cells do not exist in tight groups, but as solitary cells, but they do express these T2 bitter taste receptors, and they express the rest of the initial machinery that's required for taste signaling. So a G-alpha protein that couples to these G protein-coupled receptors called gasteucin, and a proximal signaling pathway. They also express this channel, TRPM5, which allows them to obtain sodium. Cells are marked by a few other pieces that we won't have much time to discuss today, but one is something called DCLK1, which is a marker of these cells. It's a serine-threonine kinase, which regulates traffic on microtubules, and also choline acetyltransferase. So this is an enzyme that's important and required for generating acetylcholine. This is something we usually think about having in the nervous system. And interestingly, brush cells, when they are activated by apical inputs, can secrete acetylcholine on their basolateral side to activate nerve fibers. So this is thought to be a way by which the sensory nervous system can be activated by luminal contents. So very interesting aspect of innate immunity that where there's a lot more to be discovered and worked on. So in the nasal epithelium, up until the past recent years, what was really known about these tufter brush cells is that when they are activated through these bitter taste receptors, either by kind of chemical agonists for bitter taste receptors or by bacterial quorum sensing molecules, these cells would get a calcium flux, have sodium ion exchange, and release acetylcholine. In the upper airway, this has an important role in activating trigeminal nerves and in reflex responses like cough. They also elicit substance P, which has a pro-inflammatory function, and activation of a brush cell and release of substance P has been shown to cause local mast cell degranulation. Calcium flux in these cells can also elicit action potentials, which could be transferred to other cells and end up in the release of things like beta defensins and antimicrobial peptides. In the trachea, a similar cascade of signals is elicited, and this can cause a reduction in respiratory rate. These cells came to the attention of immunologists when 3 papers were published in 2016 that showed that these brush cells not only had this neurologic function, but that they could also generate IL-25. And because of their IL-25 generation, they could activate ILC2s, and ILC2s, of course, make IL-13, which would go on to cause goblet cell metaplasia in the intestine. Shown here, so we usually think about goblet cell metaplasia as bad because we're thinking about asthma, but goblet cell metaplasia is a very important part of innate immunity in the intestine, where the mucus that's secreted and then the sweeping function in the epithelium, the muscular sweeping function, is important for getting rid and clearing parasites. Shown on the bottom here is the murine intestine that's been infected with a helminth called N. brasiliensis. And what you see is that after a worm infection, as expected, there's goblet cell metaplasia. So that's shown here with all these very dark dots in the intestinal mucosa. But if you delete interleukin-25 specifically only from brush cells, you get rid of this goblet cell metaplasia. So it's very important local immune action of the tuft cell in modulating mucosal responses. So Laura Bankova is a pioneering scientist at the Brigham. She was previously in my lab and has now set up her own lab. And she was studying the mouse tracheal airway. When reading those papers that came out in the gut, she realized that that's what she had been studying and set out to prove it. And here this is just showing you in the murine trachea these brush cells that are marked by DCLK1, this alpha-gusteucin protein, and in a fluorescent reporter mouse that you can detect them in the airway. She was able to show that these cells have interesting transcriptional profiles, and including that they have transcript for IL-25, less so for TSLP and for IL-33. And at a protein level, using a fluorescent reporter mouse, she was able to show that, again, these DCLK1 positive brush cells generate IL-25 and are the only IL-25 generating airway epithelial cell in this context. When she could harvest these brush cells, she developed an assay, a flow cytometric assay, in order to pull them out from tissue and look at their transcriptome. And this is just showing you that the transcriptome of a brush cell is quite distinct from all other epithelial cells, which are here, and is quite distinct from CD45 hematopoietic inflammatory cells. When she further looked at them, you can see thinking about differential expression in these heat maps, that there is just an extremely significant difference between the transcriptome of brush cell and other epithelial cells. As she started to compare brush cells that were pulled out from different tissues, so this is the tracheal brush cell that I've been talking about thus far, the nasal respiratory brush cell, and the olfactory sphenoid sinus area, she could see that, again, the transcriptome of brush cells shown here on the right is quite distinct from that of the rest of the tracheal and nasal epithelial cells. And when she started to look, And when she started to look at what made them so unique, one feature stood out, which is that the brush cells expressed transcript for enzymes that are required for leukotriene generation. Because we understand that leukotrienes are important pro-inflammatory factors in asthma and also can cause bronchial constriction, she followed up on this. And she developed, She developed assays where she could extract these cells from the nasal epithelium and stimulate them in culture. Here she can show you on the left that they're in ex vivo stimulation. She can show that brush cells generate CysLTs at a relatively high amount, and that is not a feature of other epithelial cells. She could also show in vivo that if she took allergen, she could elicit cysteinyl leukotriene generation in the nose with a murine allergen challenge, but she did not recover leukotrienes from the nasal lavage of mice that were deficient in brush cells. There we go. With repetitive allergen challenges, such as the fungal allergen Alternaria, Laura was also able to show that CD45 inflammatory cells that come into the lung, eosinophils that come into the lung, and ILC2s that expand in the lung were dependent on the IL-25 coming from brush cells because they were blocked by IL-25 blocking antibody. She also showed in work that we're not going to talk about today that the inflammation and expansion of these cells was modulated or inhibited by a leukotriene receptor called OXGR1 or CysLT3. One remarkable feature, many remarkable features of her studies, but one of them was really that although these cells are quite rare, shown here in the naive nasal mucosa, after allergen challenge, just a single challenge, several days later, you can detect expansion of brush cells, shown here. And you can see that they're in these clustered conformations, suggesting these are kind of newly generated cells that derive either from replication or from a local progenitor. Shown here on the bottom is just that this is not something that's unique to this allergen Alternaria. You could take another allergen like dust mite and show that you can drive brush cell expansion. So the airway can remodel in a way that we think is quite important. So just to explain now, of course, I haven't talked about human asthma here, and these cells are tricky, and we won't show any data from that today. But it is worth noting that Noam Cohen's group has shown in fungal sinusitis, shown here in this paper, in a patient who has allergic fungal sinusitis, over on this side, that despite the fact that in the inferior turbinate mucosa, shown here, that there is not much IL-25 and not many brush cells detected, in this area of focal inflammation, there is an expansion of IL-25 producing cells, and clearly some of them have the shape which are classic for brush cells. It's worth noting also that in other tissues, such as the murine intestine, when challenged with a variety of helminth or protist infections, these brush cells can expand quite rapidly. So we think that this may hold in other tissues and potentially even in the asthmatic lung. So in summary, in this part of the talk, I wanted to just summarize by saying that we think epithelial cell diversity and plasticity is much greater than we previously appreciated, that these unique brush cells have some distinct sensing and effector programs, that they generate IL-25 and leukotrienes, both of which have potential important functions in asthma. These brush cells drive both type 2 inflammation and can cause epithelial remodeling, and there I mean goblet cell metaplasia. And that although they're naive in the airway, their numbers increase in type 2 inflammation elicited by common allergens. So we look forward to finding the key areas where we think they might be important in human disease. So with that, I'm going to switch to another piece of news out of the lab, and this is on a different cell type, and this is human data, and this is on basal cell expansion and function in type 2 inflammation. So this is a work done out of a fantastic collaboration on chronic rhinosinusitis with nasal polyposis. As most of the audience likely knows, nasal polyposis is a type 2 airway inflammatory disease. Patients will get chronic nasal congestion, poor nasal airflow, loss of smell, and reduced taste. Nasal polyposis is basically an outgrowth. Polyps are an outgrowth of the sinus mucosa that comes into the nasal airspace, blocking it and causing other things like recurrent chronic sinusitis. We are very fortunate at the Brigham to have a AERD center that is spearheaded by Tanya Laidlaw, and a nice collaboration with her group and with Josh Boyce, who is, of course, the head of our division and has the program project PI for a U19 that studies inflammatory circuits in AERD. And through this group and a collaboration with Alex Shalick's lab, we've made some kind of interesting observations in the airway epithelium in polyposis. So what I'm showing here on the left is another one of these t-SNE plots from single-cell RNA sequencing, and I just want to focus a little bit more on this. This specific paper is the first of a few out of this collaboration, and it was spearheaded by Dan Dwyer and Jose Ordovas-Monten, who at that point was in Alex Shalick's lab. These spectacular junior investigators, kind of rising stars in immunogenomics, now each have their own labs, one at the Brigham and one at Boston Children's Hospital. Shown here on the left is the single-cell RNA sequencing from biopsies from 12 patients with chronic rhinosinusitis. 6 have polyp and 6 do not. What I'd like to show you as we walk around this little plot here and look at the landscape is that you can recover lots of different inflammatory cells from these tissues. You can see that we see T cells, we see B cells, we see mast cells, and myeloid cells. And you can also recover stromal cells like epithelial cells, fibroblasts, endothelial cells, and a very large group of epithelial cells in all kinds of blue here. Shown here on the white is a wider image of what I showed previously, on the right, of what I showed previously. And this is the same map, essentially, but recolored to understand where the patient samples came from, whether the disease was chronic rhinosinusitis or that with polyposis. And what I'd like to point out is a few things. Clearly, in patients who have polyposis, it's easier to recover T cells, and it's easier to recover B cells and myeloid cells and mast cells, which is consistent with these being a very aggressive inflammatory disease and kind of not surprising. What has surprised us is that when we looked at the stromal populations, when we first took a look at these plots, was that cells like fibroblasts, although there were, of course, some expansion of fibroblasts that were able to be recovered in the polyp group, what you can see is that the transcriptional signature is distinct. These don't overlie each other, but are quite distinct. We didn't really see that with the immune cells at this global landscape look. And when you come over here to the airway epithelial cells, I'll tell you that this big cluster over here in dark blue are all basal cells derived from polyp. But these in the green are derived from CRS SNP, or without polyp, and these are from patients with polypoid disease. So there is a completely distinct transcriptional signature in disease that was fascinating to us. So, of course, we started to look at what are the genes that distinguish these groups, and we could draw several themes, and I'll just comment on one or two here. When we look across secretory cells, we can see that there is a loss of antimicrobial and protective factors that are in the mucosa of patients with polyposis. So these are quite diverse factors. It could be anything from COX-2 or antioxidants, dehydrogenases, or it could be antiproteases like the antiprotease SPINK5, which is lost and is also known to be lost in other type 2 inflammatory diseases. But we could also see that in polyp, again shown here in purple, we had upregulation of some kind of classic type 2 signatures that many clinicians will recognize, including periostin, ALOX15, SERPIN-B2, and CCL26. And we had upregulation in polyp of some features that are present in wound repair programs. So quite distinct different genes. I do want to say, because this is polyp and not asthma, that some of these same themes are clearly detected in asthmatic tissue and in cells derived from asthmatic patients. This is one of many examples, but this is a recent paper by Max Seibold out in Denver, where they took respiratory epithelial cells from the bronchi, grew them out in air-liquid interface cultures, which are shown here. This is a classic technique for studying airway epithelial cells. They were control cells, or they were stimulated with IL-13. And then he did single-cell RNA sequencing. And what is shown here is just that in addition, if you look across all of the genes that were dysregulated by IL-13, in addition to upregulation of mucins and MUC5AC and an entire mucin program, there was also downregulation of important molecules in innate immunity. So those two things are hand in hand, and type 2 cytokines suppress very basic innate immune functions. So if we go back to our data in polyp, we did suspect that many of the derangements that we were seeing in the airway epithelium and this altered transcriptome related to this type 2, the aggressiveness of this type 2 disease. And so Dan and Jose did an insightful thing. They took data sets where people had grown out these air-liquid interface cultures and had stimulated them with IL-13, and they developed a gene signature. So a signature or a footprint of what IL-4 and 13 look like on airway epithelial cells. And then they took that signature and they said, is this signature overexpressed in any one place? And as they started to do that and scored areas over here, you could see that patients with polyposis had a markedly increased gene signature for type 2 cytokines. Not surprising, we thought it was a type 2 cytokine disease, and this is just documenting that. And interestingly, you know, almost 1% or up to 2% of the transcriptome of these cells were type 2 cytokine genes. So, or ones induced by type 2 cytokines. If you take that signature and then you look across the individual clusters of the epithelial cells that we have, we see that it's not only that this type 2 pressure on epithelium is in goblet cells or very differentiated cells, but you can actually see it across the epithelium, including in these immature stem-like basal cells. That same gene signature was later used by a group that studied asthma, and they also found an increased type 2 cytokine signature that could be detected across many epithelial cell subsets. So basal cells are a stem-like progenitor. Basically, all of the well-characterized differentiated epithelial cells we think derive from them, either directly or through an intermediate club cell. And as we thought about this pressure that was across the epithelium, we thought to look at basal cells a little bit more closely. And it wasn't just their transcriptome that gave us some clues. We started looking at them and doing some immunofluorescent studies. And this is P63, which marks basal cells across multiple tissues. And what you can see here is in non-polyp, there's a nice orderly array of basal cells that are at the epithelial surface. However, in polyp, I think you can appreciate that these cells are quite expanded. Now, I'll say, you can find areas in polyp where it looks nice and orderly like this, but in many areas you will see this accumulation of basal cells. And as we started to think about this, we can see in both groups that basal cells can proliferate, but there's many more of them here, and what we really found was that these cells were failing to differentiate. When we can take the transcriptional profile of these cells, we can do another technique, or the computational biologists can do another technique, where they try to align up the transcriptome of these cells. We can identify it kind of like aligning bones in an evolutionary map. So if you can identify the starting point, we can say these are basal cells, and I can identify a differentiated secretory cell. This is a goblet cell that expresses MUC5AC. You can start to align the epithelial cells across a differentiation trajectory, which is the trajectory that we expect based on this biology. And If we do that, what you can see is that in patients who have sinusitis but not polyp disease, some of the clusters, the basal cell clusters, are, as expected, less mature. The intermediate secretory clusters occupy a mid-space in pseudotime. And then the MUC5AC goblet cells are very differentiated over here. If you try to find those same groups and that same differentiation trajectory in polyp, you basically don't see it. It's completely dysregulated. So there's a failed, and that's shown here. There's no beginning point where they really look like basal cells, and there's no end point where they really look like goblet cells. So we started to think about basal cell accumulation and basal cell differentiation and what this means. And while we were thinking about that, we looked carefully at the basal cells and also noted that they were really poised for pro-inflammatory function. They expressed much higher levels of transcript for all the things we care about in the epithelium, including IL-33 and TSLP. They also express enzymes that have been shown to be important by other groups, including ALOX15 and CH25H. I didn't put it in here. And it didn't escape us also that the more poorly their differentiation was, the higher we saw them produce pro-inflammatory features. So we really wanted to kind of zone in and look at that. And we started looking at all the genes that were dysregulated in basal cells. And Dan Dwyer noted, to his credit, that basal cells expressed many genes that are in the glycolytic pathway and many genes that are inducible by HIF1. So for the pulmonary folks in the group, they'll be more familiar with this, but they're hypoxia-inducible genes, and those gene sets are shown over here on the right. And what he did is he started using the same scoring system across basal cells, and he did it across patients who have the aspirin-exacerbated respiratory disease, so very severe type 2 inflammation, polyposis, which is a little bit more intermediate, and then without polyp. And what he could see is that there was an increase in glycolysis and an increase in hypoxia gene signatures in addition to the type 2 signatures. So they seemed very hand in hand. And then we started to think about what does it mean when a cell is failing to differentiate, is secreting, we think, many pro-inflammatory molecules and has hypoxia and glycolytic gene signatures. And we came to this idea of senescence. For those people who are not cell biologists and haven't looked at this, senescence is a state of cell cycle arrest in a cell that is not terminally differentiated, and it's thought to be largely irreversible. This is often described in the cancer literature, and it's associated with the induction of proliferative and anti-proliferative tumor suppressor signaling at the same time. And although in cancer it's thought to have a role in prevention of cancer, in development, this coincident signatures are very important in developing embryos and in tissue patterning. So this is a basic cell biology piece that is studied by stem cell biologists. Importantly, senescent cells are not quiescent. So these are metabolically active cells. It's not the same as turning a cell off. And they can contribute to inflammatory disease by secreting pro-inflammatory mediators. Senescent cells have several hallmarks. As stem cells become senescent, they become large and have a flattened morphology. They have increased glycolysis, which is how we ended up thinking about them, and a variety of aspects of macromolecular damage. So we asked the question, could it be that stem cells in polyposis were somehow senescent? So of course we started to look, and of course they were, which is why I'm talking to you about them today. So shown on the left is flow cytometry, where we've sorted polyp basal cells based on a variety of markers that were previously reported and that we've developed in the lab. And here we show their expression of something called senescence-associated beta-galactosidase. This is an enzyme that's upregulated in senescent cells. So, and it's considered, there are many markers of senescence, but this is considered one of the gold standards. So we saw that these basal cells were senescent. We looked for other features of senescence, and one is lipofuscin. This is a degradative products of metabolism that accumulate in senescent cells. And what we could see is, and I hope you can appreciate, is that the basal cells, as you remember, are on the very bottom edge lined up here. And what we could see is that they accumulate this lipofuscin staining, which is easily detected and displayed here. So we started to try to figure out how we're going to connect these two pieces, and it occurred to us that maybe the very, very strong signature for IL-4 and IL-13 that we were seeing in the tissues was actually driving senescence. And so we were able to extract airway epithelial cells from either nasal scrapings of normal patients, and expand them in vitro and do some assays where we just stimulated basal cells. So this is not air-liquid interface culture. So it's very different than what a lot of epithelial biologists have been doing before. But we stimulated these immature progenitor cells with IL-4 and IL-13. And what we could see is that we could easily elicit upregulation of all of the senescence markers that we had found in vivo, including senescence-associated beta-galactosidase. We wanted to convince ourselves that this might actually be senescent and that these cells wouldn't have their normal functions. So the job of a basal cell in creating the epithelium is essentially in adult life is to repair itself. So they're the stem-like population, and people have studied them frequently in wound healing models. So shown here is our version of the wound healing model. So we take basal cells out of people, we grow them up in a dish, and once they're confluent, we scratch the center of the dish. This is called a classical scratch assay, and here is an example of a scratch that I tried to outline to make it more apparent. And then we wait for 18, 24, 48 hours, and we'll see that these basal cells grow into the wound. If we were to wait for longer, they start differentiating more and more. And so this is what we call normal wound repair, and that's shown here at 18 hours. You can see that the cells have both migrated in and started to expand or proliferate, and they're starting to close this gap or close the wound. This is just one example of what we see if we pre-treat the basal cells with this type 2 cytokine that we think is inducing senescence. We see that they now fail to close wounds. So in summary of this part of the talk, we see really extensive remodeling of the airway epithelium in chronic rhinosinusitis with polyposis. We see gain of pro-inflammatory functions and loss of antimicrobial and protective functions. We can see both in asthma and in polyposis that there's this imprint of type 2 cytokines. And in polyp, particularly, we have basal cell expansion and aberrant basal cell differentiation. We are in the process of trying to understand what senescence means for basal cells, what it means that you would have an aged or dysfunctional stem cell on the surface of the epithelium where you need certainly healthy ones, and you need on a regular basis to be able to regrow epithelium and respond to wounds. And so we're very anxious to see how this plays out. I will note that we have not generated this data, but other people have looked for senescence in both asthma and in COPD, and though the reports are rare, some of them are quite beautiful, and it's very clearly detected in both, and it increases over control. So with that, I would just conclude that what I think we've seen over the past several years is the extensive airway epithelial remodeling that can be seen in asthma, that can be seen in other type 2 inflammatory diseases, including nasal polyposis, and that we see when we model this in murine models. I think we are, with the advent of new technology, in particular single-cell RNA sequencing, instead of staring at goblet cell metaplasia, we're actually able to ask questions about the molecular architecture of the airway epithelium and what it's remodeled. I think that hopefully our brush cell example will convince you that if you change the composition of the airway epithelium, there's some very meaningful functional differences that can both impair kind of normal wound responses, but also could lead to additional pro-inflammatory functions. So I think it's been exciting so far to deconstruct classic allergic asthma, or to think about deconstructing classic, type 2 high nasal polyposis. But obviously, the real value of some of these is to try to use this technology to look at the molecular circuits that are in diseases that we don't really understand. And of course, chief among them for likely those in the audience is non-type 2 high asthma and severe asthma, and using this to deconstruct the heterogeneity that we see there. And lastly, I think there are other groups that have done beautiful work in taking airway epithelial cell signatures and trying to use that to try to identify patient populations who are responders to different therapies. Obviously, that is how we ended up with dupilumab and being able to really identify type 2 high asthma. And I think these kind of technologies will have a lot of therapeutic implication as we can bring the signatures that we're now finding that are beyond type 2 high or in type 2 low coming soon to bear. So there are a lot of fruits. And with that, I just want to close and say thanks to the people who are doing the work. So the projects that I outlined here today were really spearheaded by Laura Bankova for the brush cell work and by Dan Dwyer for the basal cell and nasal polyp group, along with, of course, our collaborators. And both of those two are really flourishing. Our current work on basal cells is being done by a collection of people in the lab, including some data that you've seen here today from Niels Hallen and from Sachin Samujawal in our group. And I'd like to say thank you for the funding and for your attention. Yeah, I'll put this back on and maintain my 6 feet.
Thank you. And I have to share with you that we had some technical difficulties broadcasting, so I don't know how many questions I'm going to receive from afar, but I did want to ask you about, it's so exciting, such exciting work, and we'll never look at the epithelium quite the same again.
Hopefully you will.
And I was wondering two things. One about these novel brush cells. Do you have a sense of their distribution along the tracheobronchial tree? Are we talking about into bronchioles as part of their discovery?
Right. So I think the classic, this is a family that hopefully is a little clear, is a little bit heterogeneous. So it's a family of cells. And some of them that are, I would say, more classic are most easily recovered from trachea and proximal bronchial tubes. However, there was actually a NIH report from many years ago about the pulmonary brush cell and about a cell which is remarkably similar, but we don't know how similar yet, which is in the rat alveolus and which has been detected around human alveolus. There's also some beautiful work coming out of UPenn and a few other labs, Columbia, and whatnot, that shows that brush cells expand in other situations, including in lung repair for things like from influenza and pneumonia. There, we're talking about the distal lung. So I think the jury's out a little bit, really, to understand exactly what a brush cell looks like in the distal lung, and certainly in the human lung, we really don't have that. One bugaboo with these cells are they're very tightly adherent to the surrounding cells. So we haven't, and they're at least at baseline infrequent, so low in number. And so they haven't showed up on most of the kind of blinded single-cell RNA sequencing pieces, and I think will require some very particular techniques in order to recover them. Even in the mouse work, Laura pioneered. She's done absolutely gorgeous work, but it required a lot of work in order to figure out how to enrich them and get them out of tissues.
And so evolutionarily, they seem like they're right through mammalian development. Is that right?
Yeah, absolutely.
All animals. I got you. Wow.
Yeah.
And the other thing I heard you mention about the basal cell and the role in cell repair, and that it might be present changes in COPD as well, in the senescence of the basal cells as well as in asthma, although I don't usually think of COPD as high in, inflammatory cytokines and the like. But I'm wondering, this insight into the senescence of the basal cells, can you correlate it with the gross histologic picture that we see in asthma and or COPD of the epithelium? My sense was often areas of detachment of epithelial cells, sloughed epithelial cells. Is there a change that you would see in gross histology?
For a senescent cell,
Yeah.
So, so no, you wouldn't really be able to detect that at a gross histologic level. I think we would be looking for them in areas where there had been detachment and the need for wound repair. That's the area where you would want to do that. Senescence, as I mentioned before, is a normal developmental pattern. And in fact, you induce senescent cells when people study development, they might study the developing limb bud or this kind of piece, and you'll see senescent cells. And so you'll see cells that come into a tissue that proliferate, that become senescent, and they secrete factors. Some people might call them developmental factors. We might call them pro-inflammatory factors, but would secrete factors that cause cells next to them to become senescent. So that's how they pattern the tissue. But that is really, it's at a molecular level. So I think it would be hard to detect unless you do specific staining to try to see these molecular changes. I mean, obviously we can, but now only that we know what to look for. Interestingly, sometimes science makes things that are bugaboos an advantage. The staining that we showed where we can detect senescent cells now was actually staining that was previously used in order to try to quench autofluorescence that's in tissue so that we could do pretty confocal images and stuff in polyp. It's something that the technician in the group developed, and it was only later that we realized that she had to do this special technique in order to be able to see anything in polyp tissue, and that special technique actually, what it got rid of is the hallmark for senescence. So we actually had discovered it in a way, but not realized what we had. Otherwise, unless you know what you're looking for, it would be hard to see. Yeah. So.
And the last question for me has to do with the implications you mentioned for non-Th2 high inflammation and the possibilities of novel discoveries that are not being driven by Th2 cytokines. Can you expand on that?
Yeah, well, so connecting it to this story, obviously there are many approaches to that, but connecting it to this story, I think senescence is not something. I think what we've learned here is that the respiratory stem cells are surprisingly susceptible to what we think are I don't want to say innocuous cytokines, but IL-4 and IL-13 among immune groups are not the most potent and are not the most, right? It's interferon gamma and it's TNF and it's whatever, right? It causes cytokine storms and do these kind of things, right? But even with these, there is a dramatic reprogramming of the cells, and I think these will not be the only cytokines that induce senescence, so they will be induced by multiple other cytokines to pattern. And the other thing is senescence can be induced by completely different things. In cancer, senescence is or precancerous lesions, senescence is induced based on macular molecular damage that's happened. You can use ionizing radiation to induce senescence. So the fact that the fact that senescence might be detected and might be important in COPD doesn't necessarily mean it has to be an inflammatory disease. It's not always connected in that way, but can be induced by other tissue insults. It's a normal part of remodeling, and how much this will be pathobiologic here. It's be a very hard scientific problem if you thought that the very thing that prevents you from developing a cancerous lesion is the thing that tamps down growth and proliferation, but comes at a consequence and an expense. And in some patients, that's exaggerated. Then there's a lot of work to figure out what are the molecular features of that you could exploit in order to both reduce one thing, but not induce a different kind of biology that you don't want, like turning nasal polyp into cancers. You can imagine that there's a yin-yang. So, great.
Thank you so much.
Thanks, Chris.
Great.